Okay, you need to get to your section of notes which says enzymes. In fact, it looks just like this, enzyme vocabulary, and it should be the back page of your notes. Okay, so this is where we are now. All right, now, because of COVID, we're having to do things a little bit differently. So normally we would do an activity. At this point in time, I'm not sure we're going to be able to do it. So I'm just going to start the notes without the activity having been done. So these are your first 4 vocab terms where it says enzyme vocabulary. You are writing down what is highlighted in yellow for each term. I'll give you a moment to do that. Okay. If you need more time, click pause. If not, these 4 words, 100%, we are going to use a lot this semester. The word enzyme, it is a funny spelled word, E-N-Z-Y-M-E. Its alternate name is catalyst. So an enzyme or catalyst increases the rate of a chemical reaction. Rate, the common term for that is speed. But in biology, we will use the word rate. So this means that enzymes make chemical reactions, makes them happen faster. So what's a chemical reaction? Well, if you eat food, you have to digest your food. So those are chemical reactions. So you know that when you eat food, you don't have a piece of pepperoni and chunks of cheese from your pizza floating around in the bloodstream. The cheese will get broken down into the lipid and the protein that makes up the cheese into those building blocks. So your fatty acids and your amino acids. The bread will get broken down into sugars because it's carbohydrate. So that enzymes speed up chemical reaction, and they're reusable. So it's like the key you use to get into the front door of your house. You don't just use it once and throw it away. You can use it for years and years and years. It's reusable. Enzymes are also reusable. And the substrate is just a fancy name given to the chemical that an enzyme breaks down. So for example, pepperoni. Okay, so the substrate is the protein in the pepperoni, and it's going to combine with the enzyme, and the enzyme's going to break down the pepperoni into amino acids and fatty acids. So the products is the name given to what leaves the enzyme or the broken down substrate. So going back to the pepperoni, the protein is broken down into individual amino acids. So the substrate is protein, pepperoni. The product is the amino acids. Okay. And I have pictures of this. It'll be made much easier in just a moment. Give me a minute. The active site, that's where the action happens. So where that chunk of pepperoni and that enzyme meet, where they bond, that is the active site. And then the enzyme is going to break the pepperoni into the substrate, and it's going to break it down into the product, which is the amino acid. Okay, so these are your terms. Okay, now remember, this is also in the notes, you're copying down in yellow. Enzymes, we learned in our last note section, they belong to the protein macromolecule group. Because of that, enzymes themselves are actually made up of amino acids. So enzymes are proteins that break down other proteins. They break down lipids, they break down carbohydrates, they break down nucleic acids, so they break stuff down. And the enzymes will only function if their specific 3D shape is correct. So this picture's in your notes. The red thing is the enzyme. Notice you have the arrow, the enzyme, the red enzyme is still visible, and throughout this whole process, that red enzyme is still there. The active site, That's where the action happens. Notice it's this weird little three-dimensional shape. Kind of looks like a rectangle or pentagon, maybe a little house with a little piece missing. So this 3D shape, this must be correct for the enzyme to work. Now, people who have insulin-dependent diabetes, their enzyme, it's not right. So they have to inject it. That's why they inject themselves with insulin. There's a problem with the shape. So it doesn't work right. So this is how enzymes work. The substrate lands on the active site of an enzyme, and here they are combined, enzyme substrate complex. They're joined together. This joining lasts very quickly, much less than a second, and the enzyme actually breaks this thing apart. So if this is a chunk of pepperoni, that protein from the pepperoni lands on the enzyme, and then the enzyme spits out 2 amino acids. So it's breaking down the pepperoni. And the enzyme goes back to the pepperoni, keeps breaking it down, until there's no pepperoni left. And then this enzyme's no longer needed. So that's kind of how it works. And these are the notes that explain how it works. So just like in your notes, step 1, when the substrate is present, the enzyme bonds or attaches to the substrate. So the enzyme joins with the pepperoni. When bonded, the enzyme breaks the substrate into pieces. And as the substrate now products, as they leave the enzyme is ready to act on another substrate. So pepperoni comes in, meets with an enzyme. It takes a piece of that pepperoni and it breaks apart into 2 different amino acids. And then this process is going to continue until all the substrate is broken down. So when all the pepperoni is gone, there's nothing left. Now, it's kind of hard to imagine this, so I have a cartoon video. Whoops, need to get to the right one. Wow, sorry about that. And now I need to refix that. Okay, so here we go. And this is less than 1.5 minutes.
Most chemical reactions do not occur spontaneously in a cell. Instead, cells rely on proteins called enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy sources available to them. Enzymes have a unique way of kickstarting reactions. They work by binding to one or more specific molecules called reactants, or substrates. Binding occurs at a special region on the enzyme called the active site. Once the substrates bind to the active site, they form what's called an enzyme-substrate complex. As the enzyme and substrates begin to react, some of the chemical bonds in the substrates begin to weaken, causing them to link together. Eventually, the chemical reactions at the active site lead to the formation of a different molecule. This is referred to as the product. Once the reaction has occurred, the product is released from the active site. The enzyme returns to its original state and is free to react again with another set of substrates.
Okay, so that is how the enzyme works in a little cartoon version. Okay. Now let's go to next. All right, so next in enzymes act as a lock and a key. So this is just an analogy. So it's using something you're familiar with and comparing it to this new thing called an enzyme. So if you think of the key to your house as an enzyme, the functional shape, that specific 3D shape of the enzyme, is what we call the teeth of the key. And you guys already know, the key to your front door, you can't go to your best friend's house and use the key to your front door to open up your best friend's front door, because these little grooves and things, these are all different depending on the key that it has. So that's why you can lock your front door and hopefully no one else gets in because they don't have a key. The key you carry for years and years and years, so it's reusable. The substrate is like the lock of your front door. The key can only do two things to your front door. It can unlock your front door, or it can lock it. Enzymes can only do one of two things. They can break stuff down, as in your digestive enzymes that break down your food, or they can make something, put something together. And there's another set of enzymes that makes energy. So, and we learned about that. We'll learn more about that later, but we all need to have energy. And if you've ever tried to stay awake as many days as possible without any chemical help, eventually you're going to get so sleepy that you can't stay awake and you fall asleep. That is because your body can no longer keep up with the energy output it needs. So it means you need to rest. And when you sleep, you use a lot less energy than when you're awake. So your body can then build back up that energy source. So this is, again, it's just like an analogy to help you understand this diagram where your enzyme has a three-dimensional shape, and then it can break something down as in here, or it can make something. Just like a lock can be locked or unlocked. Okay, at the very bottom of that paper, why use enzymes? Well, we already know that enzymes speed up chemical reactions. So there's another job that enzymes have, and it's called enzymes lower activation energy. So what is the root word of activation? Well, it's activate. If I told you that you're getting a brand new cell phone today, but you have to activate it first, what does that mean? So think to yourself, what does that mean? That's what enzymes do to chemical reactions. You can't just buy a cell phone and start using it. You have to activate it. So you either have to get a service, or you use prepaid cards, or whatever it is that you have. There has to be a service of some kind. And enzymes, the activation energy, it's the energy needed to activate or start a chemical reaction. And this is a graph. You don't need to memorize it, but let's take a look at it. This is a chemical reaction. So this is when you're starting the reaction, this is during the reaction, and then at the end of the reaction. And then this is the amount of energy needed for that reaction to take place. So if we think about digesting that pepperoni, if you're vegetarian, think of it as tofu, okay? So you have your pepperoni, and without enzymes, this red solid line, that's how much energy it's going to take for your body to digest the pepperoni without the enzyme. And so that is from this line all the way down to your x-axis. So this is the amount of energy that's needed. Well, if you use an enzyme, that's the dotted line. So notice the enzyme is a lot closer to this zero mark, this zero line, or your x-axis, than this line is. So enzymes, they're really cool because you don't need energy for them to function. So if I go back to this picture, what causes an enzyme to work is something called hydrogen bonding. When the enzyme and the substrate join together, the bonds literally change. The presence of them being next to each other or connected literally breaks the bonds apart, and it rips them up. And so it breaks down that pepperoni into protein. Then the enzymes break the protein into amino acids. Your stomach acid, its job is to take those big chunks of pepperoni and turn them into little teeny tiny pieces so that the enzymes can get to them. So activation energy, because enzymes use bonding to break bonds, they use the interaction between the enzyme and the substrate, this little interaction right here, with the active site. It does not require energy to do that. So there's a lot less energy being used to break something down if you use an enzyme. And we'll learn about later in the next unit just how much energy you make, and it's staggering numbers. So if you've been in math class, we're talking about exponents. The amount of energy is times 10 to the 19th power. I mean, it's crazy. We'll talk about that with the next unit. But even then, if we did not have enzymes, you would literally have to only be able to eat food once every 24 hours. Because without enzymes, it takes 24 hours for your stomach acid to break down the food. So it really is significant. Enzymes really do lower the activation energy as well as speed up that chemical reaction. Okay. Now, there are extreme temperatures, and I just lost myself here. Okay, the last part of the notes. Factors that affect enzyme functioning. So, extreme temperature changes and extreme pH changes alters the 3D shape of enzymes. And you're writing down everything that's in yellow here. Okay, so if you get a fever, if you have a fever of 102, could you eat a steak with baked potatoes, broccoli, and have a nice piece of apple pie for dessert, and maybe a milkshake with dinner? Could you eat all of that with a fever? Okay, no. When you have a fever, your enzymes, your digestive enzymes, have been cooked. Literally, your temperature's so hot, you can't stop it. The enzymes fall apart. So that's why if we have a fever, we can't eat lots of heavy foods. And if you've ever been sick, when you get sick, you eat like soup, maybe some toast or crackers. That's because your body just can't digest a lot of food. Plain scrambled eggs are also good, but enzymes, stomach enzymes won't work. Digestive enzymes won't work. So if you get a fever, and let's say you actually have food in your stomach or in your intestines, you might have some diarrhea later on because partially through your digestive tract, you got a fever and the digestive enzymes died and the stuff couldn't get broken down, so now you're having diarrhea. Your system's not functioning properly because those enzymes they're gone. pH changes. This often happens with a bacterial or viral infection. That can change the pH of your stomach. It can raise that pH or it can lower it so that the enzymes that are supposed to work in the stomach fall apart and they can't function anymore. If that happens, it's something called vomit or throw up. So if you throw up or vomit, that's because there's a pH change in your stomach, or it could be a temperature change, and your stomach, the enzymes don't work, and the stomach's like, I can't do anything with it, so it's got to go, and up it comes. Now, if the 3D shape of the enzyme is altered, the enzyme can't attach to the substrate anymore. We call this shape change denature. What you might want to add next to this word in your notes is the word die. That's how I remembered it sitting in your chair. Denature means die. They both begin with D. Now, granted, enzymes are not alive, so if you're not alive, you can't die, but it's a way to remember it. And it does work, because I see students and sometimes years after they've had my class and they like, I remember, got to college and got to nursing school and I knew denature was die. Like years later, they still remembered it. So what does this look like? Well, this is an enzyme. It's a protein. It has a specific active site or a three-dimensional shape, and it only acts on one substrate. So the substrate has to have this same 3D shape. Well, this says above 40 degrees Celsius. So that's like anything above like 101 degrees Fahrenheit. So in other words, a fever in a human being. This is what happens to the active site. Notice how it's all kind of elongated and weird looking. Will this substrate fit onto this active site now? And the answer to that is no. So therefore, this enzyme has denatured or died, and it will no longer function. Okay. Now, another example, which you may have had before, you ever eaten eggs? If you just fry an egg, this is comes from a chicken. So the eggs that we buy at the grocery store come from chickens. This is the yolk. That's all the fat, the cholesterol that is needed to grow all the bird parts. And then this yellow thing here and this white thing here, this is protein. When you put an egg and you crack it like they did here and you put it on a frying pan and you fry it, there are enzymes inside this liquidy part, not the yolk, but the liquid part. There's enzymes in there. And by cooking them, you denature them because the temperature change is so great, the enzymes denature and they turn white. So when you cook an egg, you fry it, it turns white because the enzyme that breaks down all this protein into amino acids, it denatures or dies, and it just so happens it gives you a color change. Okay. Enzyme graphing. If we look, this is a human enzyme, and this is a bacterial enzyme, and this is temperature in degrees Celsius. Okay, and this human enzyme, notice the high point is the optimum or best functioning of that enzyme. And this one says it's about 37 degrees Fahrenheit, which is in the upper 90 degree range, which our temperature is 98.6. Notice how when you get over, this is about 40, once you get over 100, this enzyme denatures. When it reaches the x-axis, enzyme activity goes to 0. So this graph is saying the human enzyme works good close to 100 degrees, but once you get to 100 point something, it's gonna go, it's dead. This bacteria, however, it's a bacteria, it works at 50 degrees Fahrenheit. So that's well over 100 degrees. This enzyme works at a lot higher temperature than a human does. All enzymes of every living organism, they have optimum function in the temperature ranges of that organism. Okay. Here is another one. This one's pH though. So you have 1 through 6.9, that's your acidic pH. 7, remember, is neutral, and anything above 7 is a base. So if we look at the green line only is the enzyme pepsin. So it starts here. It has optimum or best activity at a pH of 2. Well, guess what? That's the pH of our stomachs. So this enzyme functions really good in the stomach. In fact, it turns proteins into amino acids in the stomach. But notice what happens when you get to a pH of 5, pepsin denatures or dies. And after the stomach, the food goes into our small intestines. Well, our small intestines has a pH of 8. So what happens to this pepsin when it leaves the stomach and goes into the small intestine? It denatures or dies because that's all this pH over here. So pepsin as an enzyme only works in the stomach. It doesn't work anywhere else. It is specific. This enzyme, the red one, salivary amylase, and you don't have to memorize these names. This is your saliva, your spit. It works from about 4.5, has optimum pH of around 7. That's our optimum pH in our mouth. And then it will actually work up to a little over 9. Okay. And why does it work like that? Well, sometimes we're eating eggs. Eggs have a pH of 9. So will amylase work in our mouth if we're eating eggs? Yep, it'll work. So it'll work. This range will take care of a lot of our food. And amylase breaks down carbohydrates or sugars. Takes carbohydrates, turns them into sugars. So it works at this pH range because that's what our mouth is. That's kind of takes care of a lot of the foods we eat. This last enzyme, the purple line, this is an enzyme that breaks down a particular amino acid, and it works in the intestines. And notice its pH from a little over 5, but it has an optimum pH here of 9 point something. So it's going to work in our intestines. It won't work in our stomach because the stomach pH is down here. It might work in our mouth if we made it in our mouth. We do not make this enzyme in our mouth. It's only made in the intestines. So the reason that enzymes have these parameters or where they function, it's so that if for some reason arginase gets out of our intestines and into our body somewhere else, what this is saying is the body doesn't want arginase to work anywhere but in the intestines, so that's why it has that pH range. We don't want pepsin to work in our bloodstream, so it can't because blood is 7.4 and amylase dies at around a pH of 5. So if for some reason pepsin were to find its way into our bloodstream, it can't work. And that's a good thing. So these are graphs dealing with enzymes that you will have to work on. You'll have to know how to analyze these graphs, and we'll practice. So that's it for these notes.